So one of the things that we'll do in genetics is we'll look to see how a trait is being inherited over multiple generations. And this can be especially helpful if we're in agriculture, let's say, and we want to make sure that we have cows that produce a lot of milk, or chickens that make a lot of egg. Or we're dealing with corn and wheat, and we want to make sure that we have lots of fruit, lots of grain available. Most of what we see with the pedigree analysis is rooted in or based in agriculture. Now, more recently, we'll track disease genes in humans, and we want to see how that's being passed from one generation to the next, especially if we're having children and we're concerned that maybe we would be passing on a gene that would make their life more challenging or could lead to a disease state or. And so one of the common tools that geneticists and especially genetic counselors will use is something called a pedigree analysis. A word pedigree harkening back to agriculture. Now, up here I have a fairly simple, straightforward pedigree. This could be for any organism. It could be for an organism like the dog walking around down here, Alta, or it could be for a human. Doesn't matter. We've abstracted everything. And we've got this extra layer of abstraction. So circles are females, squares are males. Where we have this line connecting a square and a circle, what we're communicating is that we have a union. These are the offspring of this union. Here we have a male who has made a union with a female from another genetic lineage, and we don't actually have any information about it, which is why it's not being shown here. These two individuals have had 4 offspring, 2 males and 2 females. What's interesting is down here in generation 3 is where we see a trait, and this is a trait that we're interested in. It's a trait that we haven't seen in any previous generation. It almost looks like this trait comes out of nowhere. But one of the things that we're trained to look for when we're looking at these pedigrees, these patterns of inheritance, is we're trained to first ask ourselves whether or not this trait is autosomal or sex-linked. If a trait is autosomal, the trait is most likely being shown by both males and females in roughly equal proportions. If it's sex-linked, then the trait is going to be shown by mostly males. So taking a look here, we see that males and females are showing us a trait in roughly equal proportions. So most likely, this is an autosomal trait. Now the next thing that we do, now that we've made that determination that the trait is autosomal, is we ask whether or not that trait is dominant or recessive. Most of the time with autosomal recessive traits, we see that trait show up infrequently. So I'm going to assume or hypothesize that this trait is autosomal and recessive. Now that I've made that determination, the next step is I have to start assigning genotypes. If I'm assuming that this trait is autosomal and recessive, these 2 individuals who are showing me the trait have to have homozygous recessive genotypes. The only genotype that will show a recessive phenotype is homozygous recessive. Now the other 2 siblings could have a heterozygous genotype or a homozygous dominant genotype. Both of those genotypes will show us the dominant phenotype. And because these individuals are not shaded, we know that they're showing us the dominant phenotype, and we have to write both possibilities. Now let's look to the parents in generation 2 and see if we can maybe narrow this down. Let's see how these parents could have made such diversity in their offspring. So both parents are showing us the dominant phenotype. So we have to write both possibilities for the parents, both heterozygous or homozygous dominant. Now let's just do a quick experimentation. Let's do some quick Punnett squares to prove to us, or maybe narrow down, what our parental genotypes could be. So let's pretend both parents had a homozygous dominant genotype. Could they have made any offspring with a homozygous recessive genotype? And the answer is clearly no. They don't have any recessive genes to pass on, so there's no way that they could have made offspring or kids with a homozygous recessive genotype. Well, what if just one parent had the recessive gene, had that heterozygous genotype? and the other parent had a homozygous dominant genotype. Could they have made offspring with the homozygous recessive genotype? So doing a quick Punnett square, we see again, there's no way that these two parents could have made these offspring. So what if both parents had a heterozygous genotype? If they both had a heterozygous genotype, could they have made offspring with the homozygous recessive genotype? And the answer is yes. Both parents have to pass on a recessive gene in order for the offspring, the kids, to have a homozygous recessive genotype. Now you might be asking to yourself, out of 4 kids, 2 of the kids have a homozygous recessive genotype. Doesn't the Punnett square say or show that only 1 out of those 4 kids could have that genotype? And that is a huge misconception that we have when we do Punnett squares. Every time we make this internal section, this internal part of our Punnett square, we're modeling the possibility for just 1 kid. So each time these 2 parents came together to make their 4 children, it was like reaching into this Punnett square and grabbing a genotype. So there was always going to be a 25% probability for a kid to have a homozygous recessive genotype. Each time they made a kid, this one got that 25% probability. This one grabbed the 25% probability. And we don't know what these kids have. We can't narrow it down because there's a 25% chance they got the homozygous dominant and a 50% chance they got the heterozygous. Since we can't narrow that down, we have to write both possibilities. Now, we could keep working our way back through the generations to see where these recessive genes came from, following them through the lineage. But for the sake of our example, we're going to stop here. We've solved the mystery. We've proven to ourselves that yes, this trait is autosomal and recessive. Let's take a look at this second pedigree. When we look, we have 3 generations of individuals again. In our first generation, we have a female who has the trait that we're interested in, and she has formed a union with this male. They have 3 children, they have 3 offspring. 2 of the offspring have the trait that we're interested in, and 1 does not. This female makes a union with this male, and again with this male, we don't have the rest of his connection, his pedigree, his family, how this trait is being inherited over there with his side of this union. But that's okay. Between these 2 individuals, they have 3 offspring in the next generation. And when we look at this pedigree, the first thing that should jump out to us is that males and females are showing us the trait in roughly equal proportions. So this should indicate to us that we have an autosomal trait as opposed to sex-linked, because again with sex-linked, mostly males should be showing this trait. So again, I'm going to assume that this trait is autosomal. And because I'm seeing the trait in every generation, I'm going to hypothesize that this trait is dominant. The next step is I have to prove it to myself with Punnett squares. Collect some of that data to show me whether or not this trait is truly autosomal and dominant. So once I've made this assumption, one of the first things I like to do when I'm assigning genotypes is start with the individuals who are not showing me the trait that I'm interested in. They're showing me what I suppose or I hypothesize is the recessive trait, the recessive phenotype. So in this case, under this assumption, all of my unshaded circles and squares are showing me the recessive phenotype. The only genotype that shows a recessive phenotype is homozygous recessive. So all of my individuals who are unshaded in this example have a homozygous recessive genotype. That seems like a lot of the work is out of the way. Now I've got to look to the individuals who are showing me that dominant trait. 2 genotypes will show me the dominant phenotype, homozygous dominant or heterozygous. To try and start to narrow this down, I'm going to do crosses. I'm going to do those Punnett squares. let's say my female had a homozygous dominant genotype. The male in my first generation has a homozygous recessive genotype. And essentially what I'm asking myself, if my female has that homozygous dominant genotype, could she have made offspring that have the recessive phenotype? Looking inside of my Punnett square, there's no way that if my female had that homozygous dominant genotype, that she could have made individuals that have that homozygous recessive phenotype. She has to have passed on the recessive gene in order to make offspring have the homozygous recessive genotype. So if my female were heterozygous, there was a 50% probability the diploid zygote in the next generation would have the homozygous recessive genotype, and a 50% probability that my diploid zygote would have a heterozygous genotype. So looking at my second generation, both of these individuals are showing me the dominant phenotype. Based on this Punnett square, I know that the only genotype these individuals could have would be heterozygous. Now, this individual and this individual, we don't know his lineage. We have to make both assumptions. He could be homozygous dominant or he could be heterozygous. So, doing the quick Punnett square combination, we know the female in generation 2 has a heterozygous genotype. If he were homozygous dominant, could he have made any offspring that have that homozygous recessive genotype? And the answer is no. This male in generation 2, even though we have no other information about his lineage, has to be heterozygous. The reason why is because we have individuals in generation 3 who are not showing us that dominant trait. They're showing us a recessive trait. Now, based on that Punnett square and the fact that both parents are heterozygous, we have to write both possibilities for our male in generation 3. So there we have it, a Punnett square where we've been able to figure out just about everybody's genotypes based on the phenotypes that we saw. Our third and final pedigree that we need to take a look at is when we have a gene that's on a sex chromosome. And remember, again, for this class, we're staying focused on genes that are on the X chromosome. Not to say the Y isn't important, but most of the time we're going to see genes, especially genes for diseases like hemophilia and color blindness. We're going to see those genes on the X chromosome. So taking a look at this pedigree, we have 3 generations of individuals. And the first thing that hopefully jumps out to your eye is the idea that we've got mostly males being affected. So when we see this, our first inclination is that this trait is sex-linked. And when we make that assumption that our trait is sex-linked, we're going to assume that the gene that we're interested in is recessive. So one of the things I like to do when I'm looking at a pedigree that I suspect is sex-linked is I want to start with the males, because I know that the males have a genotype where we have to represent the X and the Y chromosome. So here I have a male who is not showing me the trait. He is showing me the dominant phenotype. So his genotype is X, capital letter A, Y. This male in generation 2, again showing us the dominant phenotype. So he has a capital letter on the X chromosome. Then I have to show the Y to communicate that this is a male individual. All 3 of my male individuals who are affected have an X chromosome with that lowercase a, the recessive trait. So that's really half the battle to our sex-linked pattern of inheritance. Now, one question I want to ask myself when I'm trying to prove this to myself with Punnett squares is how did these males get that X chromosome with the recessive gene, that lowercase a. Remember that males only get their X chromosome from their mothers. So I'm really going to be focusing in on the female lineage here. I want to start up here with generation 1. This female, we start with our X chromosomes. She is not showing us the dominant phenotype, or she is not showing us the recessive phenotype. So one possible genotype for this individual is homozygous. The other possibility is for that female to be heterozygous. I'm going to do the Punnett square. I'm first going to start with the Punnett square where I have my male and my female. My female has that homozygous dominant genotype. And by doing this Punnett square, I'll be able to quickly see if having a homozygous dominant genotype for my female could create male offspring that don't have the trait and do have that recessive trait. So immediately I see in my data, there is no way that this pairing could have made offspring that had the X chromosome with that recessive gene. So this means that the only possible genotype for my female was that my female was heterozygous. And then what I see is that there was going to be a 50% probability my diploid male zygote got the hemizygous dominant genotype, and a 50% probability my male got that hemizygous recessive genotype. Again, we don't know anything about this female's lineage. We don't have any knowledge about that. She's showing us the dominant phenotype, so we know both homozygous dominant and heterozygous possibilities exist. Looking here to the next generation, and based on my experience from this first coupling, I know that the only genotype this female could have is heterozygous. Otherwise, there's no way that this recessive gene could be passed on to the male offspring that are showing it. I know that it was the mother who passed it on to the sons. Now what's interesting when we see this combination, I want to do the Punnett square, because we've got this unusual circumstance where we have a female showing us a trait. And there's this misconception that when we have a sex-linked, X-linked gene, females will never get it. So if my female in generation 2 has a heterozygous genotype, my male has a hemizygous recessive genotype, one of the things that we see is that there is a 50% probability our female diploid zygotes will get that homozygous recessive genotype. So it isn't that females can't show us an X-linked recessive gene. It's just that we need the mother to have a heterozygous genotype and pass on one X chromosome with that recessive gene. And we need the father, or the male, to have an X chromosome with a recessive gene in order for a female to have a homozygous recessive genotype showing a recessive X-linked phenotype. Now just to finish it out, for consistency, we know that this sibling has a heterozygous genotype, and going back to our original Punnett square, both possibilities existed for this female. So there we have it, another Punnett square where we've completely figured out, at least for almost everybody, what their genotypes are. Hopefully this helps you to understand this tool of pedigree analysis, how it's used, how we use Punnett squares to collect data to show and prove to us how those traits are being inherited, what that mode or pattern of inheritance is. This is a tool that's being used all the time in genetics. It helps us to figure out when we have dominant or recessive genes, genes that are on the sex chromosome. It's a very, very helpful tool. Don't forget to finish the practice problem that goes along with this. And please, as always, email me if and when you get questions.